So we almost got through lipids 4. Any questions so far on this? Pretty straightforward, right? And then we talked about, how a lack of carbohydrate can kind of force a result in the production of ketone bodies. That's kind of where we were last time. But we haven't talked about specific details of the actual ketone bodies, right? I think this would be an interesting, whatever late night talk show you listen to, where they go out and they talk about ketogenesis, and they're like, all right, what are ketones? Name one. The toxic molecules. What are they? They're ketones. It'd be interesting to look at this. How many of you guys know the structures of ketones? Okay, so let's look at this. One of the things we said was when we are oxidizing fat or burning fat, which we do most of the time, right? That can be our most important energy source. And I mentioned this a few times. I said, unless oxygen is limiting, we probably get 75% of our energy from fat oxidation. We should be pretty good at that. In general, that's considered to be a healthy physiologic state because we can store so much fat. So with that said, what happens is when you're increasing oxidation of fat, carbohydrate can help make sure that fat is generating maximal amounts of energy. So even if a person says, yeah, I want to, burn fat, I want to lose weight, lose body fat, it's important that they're getting adequate amounts of carbohydrate. So I've told you this before, but one of my spiels now is eat what you need. Eat what you need. And the question is, well, what do I need? I heard I don't need carbohydrate. Well, actually, you do. Not too much, but you do need some. So we talked about the interrelationship of the carbohydrates, and we said, well, when we're burning a lot of fat, we tend to make up a lot of acetyl CoA, and that acetyl CoA can go into the Krebs cycle as long as we have adequate amounts of what? Oxaloacetate. When acetyl CoA is being generated in more significant amounts, our cells can compensate by, instead of converting pyruvate to acetyl CoA, which makes sense, acetyl CoA actually downregulates PDH. So when you start getting a lot of acetyl CoA, you'll actually try to slow that process down coming from glycolysis. And so the big picture is, well, you're getting a lot of acetyl CoA from fatty acids. You don't need more acetyl CoA coming from glycolysis, coming from the bridge reaction. So that bridge reaction can go pyruvate to oxaloacetate. It kind of pushes it the other way. That's if we have adequate amounts of carbohydrate. Okay, see the way that works? So carbohydrate is kind of supporting enhanced fat oxidation. That's what we can say. That's not a very sexy or trendy way of saying it, right? It sounds better if you say, hey, fat burns and carbohydrate fire, right? They go together. So that's what's happening here. And I mentioned before, I said, so if you think about conditions where you're either not eating enough calories or you're depriving carbohydrates, then what happens is the liver does two things. The liver makes ketones, and it also enhances gluconeogenesis. All right, so during starvation or inadequate carbohydrate, which right away you would say less than what is probably a good idea? 130, right? Remember the definition or one of the defining points or items of the RDA for carbohydrate minimizes ketogenesis. So as soon as a person drops below 130, it's more likely they're going to make more ketones. Okay? That's just if you're walking around without exercise. Okay? So in those cases, starvation or inadequate carbohydrate, ketogenesis in the liver goes up, so does gluconeogenesis. And they kind of go together. The ketogenesis is going up because your carbohydrate is low, your acetyl CoA can't enter Krebs. Not all of it can. So some of those acetyl CoA molecules combine. And that's what this is showing. Actually, the next one shows it better. Let's look at this. This is ketogenesis. Acetyl CoA combines with acetyl CoA. One of the CoA groups comes off, and you end up with acetoacetyl CoA. That CoA then comes off, and you end up with acetoacetate. Acetoacetate is a ketone. That's a ketone body. So is acetone. We've heard of acetone. What's acetone famous for? Nail polish, right? It's a solvent. It's basically an organic molecule, right? And if a person is making ketones, some of those ketones can convert to acetone, and they'll actually exhale them. So that's what gives a person in ketosis, can give them like a unique signature to their breath. You can actually kind of smell it. And sometimes they can taste it almost like a garlicky kind of taste.
On the last slide, what were you saying happens when there's an excess of carbohydrates and there's too much acetyl CoA?
An excess of fatty acid oxidation, and you have too much acetyl CoA. So again, imagine that we go from, let's say there's 10 acetyl CoAs in a cell, and now fat oxidation is enhanced, so now rather than 10, we have 100. That means we need to compensate for those additional 90 acetyl CoA molecules by having 90 more oxaloacetate molecules. If that doesn't happen, then the acetyl CoA, instead of combining with oxaloacetate, acetyl CoAs combine with each other. And that's what's happening. Okay.
So you need acetyl CoA to bind with oxaloacetate, but if you can't make it from pyruvate, you make it from amino acids?
You can, but under those conditions, what's happening is now you're going to end up with oxaloacetate, but rather than it combining with acetyl CoA, the oxaloacetate goes through pyruvate kinase bypass and becomes glucose. So in that case, you've got these amino acids that are contributing to making oxaloacetate, but under those conditions, the oxaloacetate is going towards glucose rather than combining with acetyl CoA. You might have to look at this a few times, or not.
What is the acetyl CoA like becoming when they're combining?
First, acetoacetyl CoA, acetyl CoA plus acetyl CoA, acetoacetyl CoA. And then the second CoA comes off, and you end up with acetoacetate. That's a 4-carbon molecule. Now, these 3 molecules up here that have this purple circle around them, those are ketones. Those are what we call the ketone bodies. Right? So this is the state of Atkins euphoria, ketosis.
So how do you like end up with the bottom ones? Like, is
I'll
it
show
just
you.
more? Oh,
Yeah. So here's an important point also. The liver is the tissue that is making these ketones. Right? This is happening in the liver. And the liver is putting those ketones into the blood. And the ketones don't just end up in the blood. Where else are they? Some of them can go into muscle, but if you're going to measure them, what else? What other bodily fluid could you measure? They're in the urine, right? So this is a long time ago. This shows how old I am and how long this has been around. Years ago, when I first started getting into nutrition, my sister's like, let's try every fad diet ever. Doesn't listen to me because I'm her brother. I don't know anything about nutrition. And she's like, on this great new diet, wouldn't believe it. I checked this morning, and I actually have, I'm ketotic. That was the old term. I'm like, what do you mean? There's ketones in my urine. I'm like, yeah, I don't think that's good. It's great. Burning fat. Like, I don't think you need to do that to burn fat. Yeah, you do. You do, Scott. I'm like, okay, right? So it ends up in the urine, also ends up in the blood. Now, a lot of people think they're on a ketogenic diet, and they might be making some ketones, but they're not accumulating. Until they accumulate, it's not ketosis. It's ketogenesis. When it accumulates and concentrations start going up in the blood and in the urine, then it's referred to as ketosis. So some people think they're on a ketogenic diet, but they never hit ketosis, which isn't a bad thing because they're maybe minimizing some carbohydrate that are giving them too many calories, but they're not accumulating ketones. So I'd argue that that's probably better than them actually getting to ketosis.
So for diabetics, when they go into ketoacidosis, is that just because they will hit a certain level and normal people don't?
Yeah. So our blood, we'll go through that in a second, but good question. Yeah, that's when it gets complicated. Nothing. So look what happens. So the other two are made from acetoacetate. See the way this is bidirectional? These two interconvert. Okay? Now, remember, these are made in the liver, and then look what happens. This process can be reversed in other tissues. So see the way it says extrahepatic tissues? So the liver makes ketones, but can't use them for energy. Other tissues can use them for energy. So the liver makes ketones, puts into the blood. Tissues like brain and muscle can use them for energy. And they do some other things. There's a lot of articles about this. To be honest, there's the literature on benefits or effects of ketogenic diets nowhere near complete yet. That experiment's still going on. Lots of participants that aren't officially enrolled in the study, but they're doing it. But anyway, there's a lot to learn about that. I read a review article a couple months ago about it. They said it could do this, could do this, could do this. Not proof yet. So they're looking at some of the other things that these ketones do. Okay? But that's the point of that. Now let's look at their structures. What are ketone bodies? We said that these guys are beta-hydroxybutyrate, acetoacetate, and acetone. Yes, you need to know their names. You need to know how to spell them. You don't have to write their structures. Okay? But let's look at these. These are called ketone bodies. Are they ketones? What would mean they're a ketone?
Double bond.
Remember that? Fructose is a ketone. That C double bond to oxygen. So acetoacetate is a ketone. Acetone, a ketone. Beta-hydroxybutyrate, not a ketone. But it's included in here because it interconverts. So it's included as one of these ketone bodies that are produced.
What are the reasons why ketones could end up in your urine other than just ketosis?
Sure. So a better term for probably what we call the ketogenic diets is nutritional ketogenesis. There's some other things that can cause that. Yeah, even in uncontrolled like type 2 diabetes or something, ketones tend to go up in the blood. And that's diet related, but not really, willfully dietary induced. So one of the terms now is nutritional ketogenesis, where you're minimizing carbohydrate to force ketone production. But there are some other things that cause that anyway. So here's one of the issues. If you look at these guys, do they have any acidic characteristics? Yeah, they're keto acids. They've got that carboxylic acid group on 2 of them, right? So they can affect pH. They're relatively weak acids, but they're acids. In other words, as these start going up in the blood and in tissues, your tissues use their buffering systems to minimize changes in pH. So most people can buffer against the ketones. So for most people, it's not an issue if ketogenesis results in ketosis. But in diabetics, it's a little bit more of an issue because they have other issues that are also taxing buffering systems. So in diabetics, they need to be careful with too many ketones in the blood. It can drop the blood pH, and that can be a problem. If it's uncontrolled and sustained, it can become a lethal situation. Okay, so it's important that diabetics watch ketones in the blood, but for most other people, probably not a big issue for most people. Okay, again, the long-term studies underway and poorly controlled, I would say.
So then someone who's a diabetic and then is trying to go on a ketogenic diet to reduce carbs and lose weight or something, that could be dangerous if not monitored?
Yeah, again, I think eat what you need. So one of the big questions is how many grams of carbohydrate does each person need? And like that's, so the study that we're trying to start right now, that's one of the big things I want to try to get data on. How many grams of carbohydrate does a person need, and how different is that from type 2 diabetics to non-diabetics? Because really, if you think about it, you need 130 grams of carbohydrate, basically. And how many more? Depends. If you're exercising, you can support that. But so they are probably watching their carbohydrate anyway. So they need to watch, blood glucose as they're taking in carbohydrate, but they need carbohydrate as well.
Is there any data on pre-diabetics and ketosis or carbs?
I'd say that would be individualized. I haven't seen that, but that's at that point, they could still be able to buffer effectively. Mm-hmm. More than likely they are. So this is an interesting slide. So this is what I mentioned before, and we talked about this. We said, well, the liver glycogen only lasts about 18 to 24 hours, right? So let's say, for example, I'm intermittent fasting, right? Because everybody is. If we do this, let's say, for example, we skip dinner, and we go all night, and then we skip breakfast. What's happening metabolically? Well, the lower that your hepatic glycogen goes, in general, the more ketones you start making. Right? And even if you don't completely deplete it, overnight, ketone production starts going up. You're not consuming glucose. So if you look at this, though, like, if you look at this, like, overnight, and I have the data, I'm not going to hold you to that, but overnight, you could even see, like, if you measured blood ketones at night, and then again in the morning, you'd see the ketones go up a little bit in the blood. And then typically, what do? We eat oatmeal, carbohydrate, blood glucose goes up, so does insulin, glucagon goes down, ketones go down. So it does a little bit of a bump. If you don't have carbohydrate, look what happens. They start accumulating. And I said before that this is kind of a survival mechanism. So what's happening initially is our tissues, including the brain and central nervous system, still completely relying on glucose. As glucose becomes limiting, our body starts switching over. That's what we're seeing here. This is starvation. The ketones start accumulating. If no carbohydrate comes in or inadequate amount comes in to blunt ketogenesis, ketones in the blood start going up. And after a few days, they'll start to become significant enough to provide a solid energy source for the brain and central nervous system. So our brain can kind of switch over. Does our brain work pretty well on them? In general, it does. There's a few things that our brain does better on glucose than it does ketones. Some cognition, some processing stuff, but overall, it's an energy source that works for it. But if you look, there's two things I want you to take away from this, and one of the main things is, they basically start accumulating so that after probably a few days to a week, they're significant in the blood. And the second thing, look at which one's most common. It's the beta hydroxybutyrate. So the most significant source of energy from ketones is from beta hydroxybutyrate. That's the most significant form in the blood. Does it matter? It easily converts back to acetoacetate. And then what does it go back to? Acetyl CoA. So now think about, for example, what's happening here. Your liver is putting ketones into the blood. The ketones are picked up by, let's say, brain cells. And then what do the brain cells do? They revert it back to acetyl CoA. Now you've got significant acetyl CoA that's in the brain cells, in the mitochondria, that need to enter Krebs cycle. So what do they need? Oxaloacetate. Where's that coming from? Your liver's also doing what? Gluconeogenesis. So the liver's doing two things here. It's providing glucose. Some of that glucose is used by like red blood cells and some other tissues that are relying on that. But the glucose that's coming from gluconeogenesis is also supporting use of energy from the ketones. So your liver is putting glucose and ketones into the blood, and in the brain they kind of merge, where that glucose can actually support use of the ketones, acetyl CoA.
So does that mean when you're in ketosis that like alcohol would have more of a damaging effect on your liver since your liver's doing more than one process?
It could, yeah. In general, the ethanol in there is kind of one of the top priorities for the liver. So it'll tend to alter most other metabolism, and significant parts of liver cells are then using their systems to break down that ethanol. So it could be a significant impact. And the other thing here to look at is, right, these two, you can see why these two interconvert. Their structure is very similar. See the way this one's different? In order to go from acetoacetate to acetone, that's a decarboxylation. And it's unidirectional. So once you go acetoacetate to acetone, it's purged. It doesn't go back into the ketone pool. So it's slow. There's not much that becomes acetone, but it does. And that's one of the ways that you lose those carbons is acetone. Got it? So ketosis is the abnormal rise in the concentration of ketone bodies in the blood. And it can be present during starvation, a low carbohydrate diet, or diabetes, right? And with diabetes, if the glucose isn't going into cells, it's like the glucose isn't there. So fat oxidation goes up. You struggle with going pyruvate to oxaloacetate. So it's like the glucose isn't in the cells. So the carbohydrate isn't utilized. So that can lead to ketoacidosis. The ketones are acids, which can cause lowering of blood pH. Again, I think most people in this area will say for healthy people, that's usually not an issue. Certain unhealthy populations, they'll have to watch that. Good there? I think that's all I need to say about that. So that's minimal carbohydrate. Right? Minimal calories, for example, starvation. Now let's look at the opposite, where we might have significant calories, including significant amounts of each of the macronutrients. You know, how do we make fat in the body? This is wonderful, isn't it? Fatty acids can be made from acetyl CoA, right? It's basically the reverse. Beta oxidation is fatty acids through acetyl CoA. Fatty acid synthesis is acetyl CoA to fatty acids. So the substrate is acetyl CoA. And remember, we can get acetyl CoA from carbohydrate, proteins, or fats. If we're consuming too many of any of those, it can lead to lipogenesis. So the main sites for fatty acid synthesis, though, are the liver and fat cells. And in humans, it really depends, but I wouldn't say the liver is a huge producer of fat. I think probably most people won't, but it can make some. And then the fat leaves the liver and should be stored in fat tissue. But beta oxidation occurs where in the cell? Beta oxidation, matrix. Fatty acid synthesis, cytosol. Right, they're opposing pathways, but they have common players between them. So the cell puts them in different parts. Right, so in general, if beta oxidation is elevated, fatty acid synthesis, down. They're regulated oppositely. Okay? So if Krebs cycle activity is sustained and there's more energy coming in through the cell, and Krebs cycle is being downregulated because there's so much ATP already, Krebs cycle is slowed down because of those allosteric enzymes that are sensitive to ATP. So let's say I'm consistently overeating. I already have a lot of energy, already a lot of ATP in let's say liver cells, and now I continue to push more carbons into the system. Our body has a way of decreasing Krebs cycle activity because we don't need to support ETC anymore. And kind of an escape route is the citrate. So the citrate will actually leave the matrix and go into the cytosol. We've mentioned this once or twice already. And then in the cytosol, this is what happens. It's lysed, and it goes back to its original components. So in the cytosol, citrate lyase adds a CoA to the acetate to make acetyl CoA, and then oxaloacetate is formed. That takes an ATP. So citrate goes into cytosol. It's split into oxaloacetate and acetyl CoA. Let's follow the acetyl CoA. This is the steps in the synthesis of a fatty acid. I have it here. I think the newest edition has it this way as well. Again, I'm simple-minded. I like linear kind of stuff where I go, let me just follow what's happening here. So we'll go through this, and then I'll tell you the key points that you need to know. So far, big picture, we're making a fatty acid. We're in the cytosol, substrate, acetyl CoA. That's all we got to remember. Now look what happens. Acetyl CoA is converted to malonyl CoA. Have you ever heard of that molecule before? Probably not. And the only difference between acetyl CoA and malonyl CoA is one carbon. Here's acetyl CoA. See what it looks the same? We added a carbon. Carbon comes from CO2. Now we have a 3-carbon malonyl CoA, right? We're adding a carboxyl group. So the enzyme is called a carboxylase. It's specific to acetyl CoA. So the enzyme is, and you need to know this name. It'll be in the study guides. I can put it in there. Acetyl CoA carboxylase. Adds a carboxyl group to acetyl CoA to get malonyl CoA. Right? Again, takes ATP. So again, if you think big picture, is this anabolic or catabolic? We're making a fatty acid, so we're going from acetyl CoA to long fatty acids. It's anabolic, and that takes energy. So far, it's 2 ATP to do this. All right, we're investing. It's pretty efficient, though, if we watch this. So now what happens? In your notes, put this. See these little boxes here? So in your notes, like put an arrow or something and say, this box right here is part of fatty acid synthase. Fatty acid synthase. It's part of a complex. You ever heard that, fatty acid synthase? Makes sense though, right? This is the enzyme complex that synthesizes fatty acids. So all of these are different steps that take place on fatty acid synthase. Right? Remember, this is in the cytosol. And you don't have to remember this, but CE is condensing enzyme, and ACP is acyl carrier protein. Those are essentially two parts of fatty acid synthase. The most important parts for us to look at. All right, so that's big picture. And so what's happening along here is we're basically adding carbons until we get a fatty acid. So let's read through this and then we'll look at the picture. Fatty acid synthase is the enzyme that makes fatty acids. It combines malonyl CoA and acetyl CoA molecules. Well, where did malonyl CoA come from? Acetyl CoA. So the carbons are coming from acetyl CoA. Now, this cycle here repeats until you end up with palmitate. I mentioned that before. So why is there so much palmitate in animal tissues? Because when animals store fat, a lot of it is stored as palmitate. And we know that's a C16-0. It's a saturated fatty acid. Acetyl CoA only has 2 carbons, so we got to do this a few times. Palmitate can then be added to glycerol to form triglycerides. Triglyceride can be stored right in a fat cell. If I'm doing this in a fat cell, that triglyceride can just stay there. If it's in the liver, then the liver will make VLDL, put the triglyceride in there, and put the VLDL into the liver. Remember that? And then the VLDL goes to the capillaries, lipoprotein lipase, takes out the triglyceride, and that's how we make fat in the liver, store it somewhere else. Okay, so now let's look at this. An acetyl-CoA molecule became malonyl-CoA, and malonyl-CoA binds to fatty acid synthase, right here. A second acetyl-CoA molecule binds directly to fatty acid synthase, right? So at that point, we have 5 carbons, right here. CO2 comes off as these 2 molecules combine. So these 2 molecules combine, this carbon binds right here, CO2 comes off, so now we have 4. And now it's a matter of rearranging the molecule, reducing, rearranging. NADPH, sounds like NADH, but it's got a phosphate. NADH is involved in mostly energy production. NADPH, biosynthesis. So it's a source of electrons, basically hydrogens. Again, you don't have to know structures, but look what's happening. Got CH3, now we can add hydrogen here and a hydrogen here. They came from NADPH2. So that's what's happening there. So basically reduction, and then dehydration, reduction, molecules changed again, and then look what it does. It goes from this site, ACP site, it shifts back to CE. Now this molecule, instead of here, is here. Why did it do that? Because the ACP site is the one that can accept malonyl-CoA. Right? ACP is where malonyl-CoA can come in. So now another malonyl-CoA molecule is added. And then we condense, lose a carbon, and now we're here. But what did we do? We picked up 2 more carbons. So let's look at this initially, this big picture. Have 5 here, we end up with 4. Rearrange, reduce, dehydrate, add 2 more. So now we're at 6. 1, 2, 3, 4, 5, 6. How many does palmitate have? 16. Repeat this sequence. This is an arrowhead. So now what does it do? It goes through that again. Why is it doing that? So we can make a molecule that can then be transferred to the CE site so that another malonyl-CoA can be added. Carbon comes off, so we add 2 more. So at the end of that one, we're at a C8. Repeat it. C10, C12, C14, C16. When it hits C16, it's released, and you palmitate.
I'm a little confused why when CO2 comes off, why you still get 2 carbons added on.
Because malonyl-CoA has 3.
Oh.
So here's a couple points, right? One is, if you want to regulate this, where are most of the carbons coming from? As what molecule? Malonyl-CoA. So acetyl-CoA carboxylase is a major regulatory point. Right, and remember, this is in the cytosol, and this is the enzyme that is going to determine how much malonyl-CoA is available to become part of fatty acid synthesis. So you need to remember 2 things that upregulate acetyl-CoA carboxylase. One is citrate. Citrate positively affects acetyl-CoA carboxylase. Makes sense, right? More citrate, more basically substrate for the enzyme, so it feeds forward. Which hormone do you think would also increase this activity? Yeah. This is an anabolic process. One of the most powerful anabolic hormones we have, insulin. Insulin increases activity of acetyl-CoA carboxylase. And so some people seize on that and say, don't eat carbohydrate because it leads to fat production. Kind of. Depends on a lot of other things. Okay? Right? So this normal process, it's a way that we can convert some of the things that we might not need into an energy source and store it that we can use later. So again, remember all of this is kind of in the balance of what the overall picture is of the balance between our energy expenditure, intake versus expenditure on calories. Okay? Any questions on that? Uh-huh.
I guess just like which steps exactly you want us to know and the names, because I'm just I feel like the diagram on the right is kind of confusing.
So the diagram on the right is the binding sites of fatty acid synthase.
Okay, so it's like the malonyl-CoA is coming in to bind to the synthase?
Yes.
And the synthase has the CE and ACP sites on it?
Yes.
Okay.
Yeah. So that when this starts, you need an acetyl-CoA and a malonyl-CoA. All the subsequent cycles, just malonyl-CoA.
So that very top one is acetyl-CoA being changed to malonyl, and then that's just in the ACP site until it gets enough carbons?
Until the structure of the molecule changes to a form that is then recognized and moved to the CE site. Right, it's here, combines here. Now it's ACP, stays on ACP, it's reduced, dehydrated, reduced, and now it can move from ACP to CE. That's what's happening here. Was here, now it's here. Now this is open. This is open so that another malonyl-CoA molecule can bind, and that's what's happening here. And then once again, they condense. So nearly all the carbons are added as malonyl-CoA. That's why that's such an important regulatory enzyme. So you can imagine after you eat, Blood glucose goes up, so does insulin, so does fatty acid synthase activity. When you're fasting and insulin is low, fatty acid synthase activity down. Right? That's when we're burning fat, not storing it in general. So you can look through that, look at the points I make in the study guide. If you have questions about this, let me know. But let's move to protein, right? Had enough lipids? So here's protein lecture 1. And there's parts of this we'll go pretty easy on, but here's the overview. We'll talk about dietary protein as a requirement, like why do we have to have it. And then we'll talk about digestion and absorption, and then we'll talk about whole body amino acid metabolism. And I'll just make a few points on that because obviously it's pretty complex. So here's a couple questions for you. Why do we need to eat protein? Is it essential? Remember I said if you're on Survivor, water, then protein, probably your 2 targets. Form whatever alliances you have to. Okay? I think they still form alliances, I don't know. Right, so what are 2 reasons you think we have to have protein? Yeah, specifically, what's in that protein that we need? More specific? Essential amino acids. So 2 reasons. Because dietary proteins serve as a source of essential amino acids. And then I think you had the other one, Sarah. Nitrogen. Right? That's what makes protein different than lipids and carbohydrate. Nitrogen. So why don't we just eat dirt? Right? There's quite a few microorganisms that can take elemental nitrogen and break it apart. Right? It's usually got 3 covalent bonds that are linking the 2 nitrogen atoms. We can't do that. But some microorganisms do. They can break those nitrogens apart, assimilate it into different molecules, and then we can consume that. So our most significant source of nitrogen by far and away, protein. And it includes nitrogen. So those are the 2 reasons we have to have it. Source of nitrogen, source of essential amino acids. How many amino acids are used during translation to synthesize proteins in humans? 20. 20. Right, for us, there's tRNAs for 20 amino acids. How many of these are essential? 9. That means have to be in the diet. So remember what essential means? We require them. We require a nutrient, but we can't make it or make it in adequate amounts. So we actually make most of the essential amino acids, just not in adequate amounts. There's only 2 or 3 that we don't make at all. Okay, and here they are. You can say essential, or you can say nutritionally indispensable. Okay? And here they are. Yeah, you got to remember those. So make a, whatever you have to do, a mnemonic device or whatever, right? Private Tim Hall, I think, is the one that's the cheat sheet on this. Private Tim Hall, phenylalanine, valine, threonine.
See that? Oh, wait, just kidding. I was looking at like the start code and I was like, well, people that are like don't eat enough protein, could it just like down-regulate their ability to do like transcription and stuff? But that's not what I'm reading.
So I have the top 3 up there for a reason. Leucine, isoleucine, valine. What are they? They're like a subcategory. They are the branched chain amino acids. Have you ever heard of those? Those are magic, right? Hey, take branched chains. Yeah, I eat chicken, right? So the branched chain amino acids are the top 3. And because literally their structure has a branch in it. And that's why they're called the branched chains. So the branched chain amino acids, the 3, they're all essential. And so that can have an effect on quality of diet. We'll talk about that next time. Okay? Of those three, leucine's very unique. So I want you to remember that leucine is not only used for translation, leucine is an anabolic signal. Do you guys study tripaoperon or anything in biology? Lacoperon, how some organisms basically sense a healthy environment for growth based on a constituent that they have to have, and then it signals anabolic phase and replication. So different organisms are responding to different things. For us, in protein, kind of leucine's that one. So leucine increases protein synthesis, decreases protein degradation.
Can you repeat the second thing that leucine does besides translation?
It's a signaling molecule. Yeah. Signals anabolic phase. And we won't go into this, but the leucine, and this is really exciting. You guys probably, I'm sure you heard about it. Some of the metabolites of leucine bind proteins that are involved in this, and for years they went, leucine's a signaling molecule. Nobody could find out what it bound to. And then about 2 years ago, a group in New York, Sabatini's lab, I think it was, found the protein that's actually binding to one of those leucine metabolites, and that's kind of the holy grail of how leucine actually works. So if you look at that leucine signaling pathway, it kind of runs right along insulin. And then they converge. So one of the questions is, so it's basically kind of stimulating growth like insulin, but it's kind of on the side. So one of the questions is, can you modify a person's diet to compensate for compromised insulin signaling by giving them more leucine? That's kind of one of the big questions now, and you might be able to. So we'll talk about protein quality next time, but a lot of the protein quality dependency is on leucine, how much leucine is there. It's also a reason why there's some evidence to suggest branched-chain amino acids are really important. Do you need to supplement with them? Probably not, as long as you have a high-quality protein in your diet. Okay? They also are unique because they can be an energy source. We'll talk about those too. Okay? And then the other ones are required for translation. So here's one of the issues, like how much protein do we need, and what happens if we go too low? So remember, the genetic code determines a protein's amino acid sequence. And what if the genetic code, though, specifies for an essential amino acid, and that essential amino acid is not present? So what happens to protein synthesis? Stops. It's limited, right? Remember, during translation, that ribosome is moving along the mRNA and it's pulling in the appropriate tRNA to make that match. Now, what happens if a person doesn't have adequate amounts of essential amino acids in their diet? Translation's occurring, that tRNA might not be loaded with an essential amino acid. Translation's kind of all or none. You either run all the way through and make a complete protein, or you don't. So you can significantly impair protein synthesis, and there's tons and tons of evidence for this, right? We've seen lots of graphic photographs, for example, of people that are starving or have starved, and you can see the way they've wasted, and basically it's a lethal condition because protein synthesis stops. And it's not like just all of the amino acids that we have, it's not like we can continue to reuse them. We can do that some, but not enough. We have to consistently take in essential amino acids to basically support protein translation, or it's life-threatening. And so that's one of the reasons why. So one of the questions is, what about these amino acids? We've all seen this, right? You've heard corny things people say, well, the only thing different about the amino acids is their side chain. So we know all that, and we know you guys are going to draw a couple of these. I'll go easy on it, though. All amino acids have the same core structure. Their side chains is what makes them unique, right? So 20 different side chains. And if we look at the top left, or right over here, remember this is glycine. Its side chain, just a hydrogen. So glycine's the simplest amino acid. Is it essential? No. Alanine, remember we talked about that? Alanine can be made from pyruvate. Is it essential? No. Pretty simple structure. Just that methyl group is in its side chain. And then we have aspartic acid and phenylalanine. Phenylalanine is essential, right? Look at its side chain, hydrophobic ring, methylene bridge to a hydrophobic ring.
Is that the one that has an allergy, or am I thinking of something that just?
No, you're right. We'll talk about that. Yeah, PKU and right. Remember, it's the interaction of the side chains that determines the overall protein structure. That's why, if you think about it, we can make so many different proteins because along that protein chain, there's 20 options at every site. It's not like DNA or RNA, you got 4. And they really don't have that much of a difference in structure to make the difference a protein does. So that's why we can make so many different proteins. Have you ever heard how many different proteins we probably have in the human body? Yeah, but different types, probably 30, 50,000 different ones. But, you know, all of those possibilities because we have all these different interactions between the amino acids. So we're going to go, I want you guys to be able to draw peptide bonds. How many of you guys have done this before? Okay, so we're going to do it a little bit.
So do you want us to know how to draw the 4 that were on the previous slide?
I'd love that, but you don't have to. I'll tell you which ones.
Okay.
Yeah. Because you'll thank me when you see like leucines. Yeah, you'll go, oh. Okay. So peptide bonds are covalent bonds, and probably the main guy that figured this out was a guy named Linus Pauling. Have you ever heard that name? Wow, nothing. Linus Pauling is the only individual that has individually won 2 Nobel Prizes, one in medicine and one in peace. And a couple interesting things about Linus Pauling. One is genius in this kind of stuff. And he actually applied for a position at Cal Poly a long time ago. He was an Caltech faculty member, professor at like 21. Yeah, genius, right? But very controversial. Remember that one of his Nobel Prizes was in science, and he worked on covalent bonds and everything. And another one was he was anti-nuclear weapons proliferation. And he was very, very adamant, very outrageous in all the things that he was willing to do and say to try to stop that. So this is like in the 50s and 60s Cold War. You guys don't remember the Cold War. So when I was growing up, the Cold War was just kind of ending, and all of that stuff happened. This is when you had bomb shelters and all this stuff. Well, he was also trying to figure out the DNA structure. Remember that? Remember Rosalind Franklin, who probably should have got credit for it, but didn't, and it was Watson and Crick. Linus Pauling thought it was a 3-part strand instead of a double. There was one conference in Europe that the United States wouldn't give him a visa to go. They said, if you go, you're not coming back. And he didn't go and see the data. And remember that X-ray crystallography film where you could look at that and go, oh, there's your double strand. He didn't see that. Otherwise, he would have figured that out too, probably. And he applied for Cal Poly, and they didn't give it to him because he was too controversial. So he was going to be a chemistry professor here, and they're like, The rumor is Phil Bailey got it. You ever heard of him? Dean Bailey, who was here for probably 30 years, 40 years maybe. He's the reason the new chemistry buildings are up. That was Warren Baker, the old Cal Poly president, and Phil Bailey are the ones that got all that together to make that new building. So Bailey was here for a long time. So overall, I'd say, I think that worked out. Pauling actually lived up in Cambria for years. And I remember when I was an undergrad, I was grading for my instructor, and I walked into her office, and she was kind of rattled, and she's like, oh, she'll never guess who I just talked to. Linus Pauling. I'm like, you guys. I don't know who that is. Play sports, because and she's like, Linus Pauling, 2 Nobel Prizes. I'm like, nothing. Chewed her out. Because Linus Pauling later on in his life was in vitamin C. And that's where a lot of people think he got it as a Nobel Prize. She was on the local news talking about how don't overdo vitamin C, and he was a huge vitamin C advocate. So he saw it on the news, called her in her office, and chewed her out. She's like, I've never been chewed out by a Nobel Prize winner. Whatever. So anyway, number 3 here, what are we doing? So he was around the area for a long time. He lived to be probably 90. Yeah, and he used to, I'm not doing 329, but in 329 lecture, I used to talk about the amounts that he believed we should take and why they probably were way too much. And he had some interesting theories about that. So he got into nutrition. He's a smart guy, right? So of course he ended up in nutrition. So he was the one that kind of figured out these peptide bonds and how they interact and how they're formed. So remember, the peptide bonds are formed between an interaction between the carboxyl group on one amino acid and the amine group on another. When that covalent bond is formed, a water molecule is removed. So when you draw a peptide bond for me on the final exam, make sure you incorporate that water molecule coming out. And that forms this peptide bond. So you're going to be able to draw 3 different amino acids separately. You're going to be able to draw those 3 amino acids linked with 2 peptide bonds. The amino acids that I want you to be able to draw their structures are glycine, you're welcome, phenylalanine, and tyrosine. Why am I choosing those 2? PKU. We'll talk about their metabolism. So those 3, glycine, phenylalanine, tyrosine, draw them separately and in peptides. Now, this shows that at neutral pH, the amine group is charged. Remember your formal charges? This nitrogen is in 4 bonds, so it has a positive charge. And the carboxyl group is negatively charged, so make sure you put your charges on there, on your ends. Remember the amine group here, if that's open, that's your N-terminus. And the other group is actually, the other end is your C-terminus. Just means the carboxyl group is open. Went through that, right? Let me do one more slide. Now, in a lot of your other classes, you've probably talked about a lot of this stuff down the left side. So this is the physiological rules for protein and amino acids, right? We say, oh, proteins are enzymes, antibodies, blah, blah, blah. Talked about all that, right? We're going to talk about some other things in here. We're going to talk about the actual metabolism of the amino acids, like what happens to them. How are they made? When they're broken down, what happens? And so we're going to look at, for example, how you can get energy out of the amino acids, some of them. We've talked about that a little bit. We'll look a little bit about gluconeogenesis, and then we'll talk about synthesis of other key molecules. One of the points here is protein is a source of nitrogen. In order to get that nitrogen, we have to pull the nitrogen off the amino acids. So there's lots of reactions where that's involved. So that's a good thing. The problem is accumulation of nitrogen can generate ammonia. I think we all probably realize ammonia, not good for us. So we'll talk about how our body actually takes that nitrogen and puts it and excretes it in a safe form, and that's urea. So we'll talk about urea production and the urea cycle and how that's involved in safety and amino acid metabolism coming up. So that's probably the unique part of all this that we'll look at. And again, we'll kind of go easy on. I'll try to make some examples to give you an overall idea, but not go too heavy into it. And then that will set us up for the last protein lecture, which is protein lecture 2, where we'll talk about requirements for protein in the diet. Okay? Okay, guys, thank you. See you Thursday, if not before.